Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs June 2023 Shift 2. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. These are solved questions of part C. If you are new to my channel, please do like, share and subscribe. I have also uploaded old CSIR PYQs. You can refer to my channel for the same. Coming to part C questions, the first one. The following are some statements made regarding mutations. Which one of the following options represent the combination of all incorrect statements? Change of DNA sequence from AGC to ATC in non-coding strand can have major impact on protein production. Now AGC is UCA on the RNA and ATC is UAG on the RNA. So UAG is a stop codon. So if you have stop codon coming in between, then it is going to have major impact on protein production because incomplete protein will be produced. So A is correct. Then suppressor mutation restores the original phenotype only when a second mutation occurs at the original site of the mutation. So suppressor mutation, it can occur at a distant site in the same gene as the first mutation, or on different genes, or on even different chromosomes also. Now what they have told is suppressor mutation restores the original phenotype only when a second mutation occurs at the original site of the mutation. So a second mutation occurs at the original site of the mutation is when there is back mutation occurring. So it is not suppressor mutation. So B is an incorrect statement. Mutation rates remain the same in all organisms. It varies significantly among different organisms. So C is also not correct. Strand slippage during replication is a consequence of loop formation in one strand of DNA. That is also correct. Hydroxyl amine adds hydroxyl groups only on cytosine. That is also correct. So they have asked for all incorrect statements, and B and C are the incorrect statements. So your answer is option 2, B and C. Then next one. An investigator identified a nuclear localization signal at the C terminus of the protein X. To analyze the localization of protein X, the investigator fused protein X with GFP at the C terminus. The fusion protein was detected in the cytosol. When the nuclear localization signal was fused with GFP at the N terminus, the NLS tagged GFP extensively localized in the nucleus. Based on this observation, the investigator made a few hypotheses. Which of the following option gives the combination of all possible hypotheses that best explain protein X trafficking mechanism? Now if you see the photograph I have given over here, you will come to know what they have asked in the question. An investigator identifies a nuclear localization signal at the C terminus of protein X. This is the N terminus, this is the C terminus. At the C terminus there is an NLS and this is your protein X. Now he wants to analyze where the protein X is getting localized in the cell. So to analyze localization of protein X, the investigator fused protein X with GFP at the C terminus. So this is protein X, this is N terminal, this is C terminal. So next to NLS, that is he fused GFP. The fusion protein was detected in the cytosol. It was not localized to the nucleus, but it was found in the cytosol. When the nuclear localization signal was fused with the GFP at the N terminus, the NLS tagged a GFP extensively localized in the nucleus. Now when the GFP was tagged to the N terminus, it was found in the nucleus. Based on this observation, the investigator made a few hypotheses. Now which is the best hypothesis you have to find out. The first one, the basic amino acid stretch in the protein X GFP chimeric construct is masked by the GFP sequence and thus could not was thus not capable of directing entry of protein X GFP into the nucleus. So in this case, that is when GFP is fused at the C terminus, GFP masks the NLS and is not capable of directing the entry of protein X GFP into the nucleus. So that is correct. Statement 1 is correct. Because GFP is over here, NLS is not exposed and the protein is not directed toward the nucleus. So that is correct. The X protein in the full length S GFP chimeric protein is post-translationally modified that impacts its import into the nucleus. That could be one reason, because X protein when post-translationally modified could have changed its conformation that have masked NLS, so its import to the nucleus got hindered. So that could be another reason. So B is also correct. Fusion with GFP makes the protein X too bulky to enter the nucleus through the nuclear pore complex. That is not a correct answer. The GFP is post-translationally modified that impacts the import of protein X GFP into the nucleus. If GFP was post-translationally modified, then NLS would have got exposed and it would have favored the protein X to enter the nucleus. So D is also not correct. So the correct options here are only A and B, and option C is your answer. Then next one. 2 batches of antibodies Q and R were generated for an antigen, and affinities of both the antibodies were assayed using pure antigen. Given below are scattered plots obtained for the antibody-antigen binding assays and the inferences drawn upon Stackad analysis. So this is the Stackad plot. This is bound and free form. This is the bound form. Now Stackad plot, it is used to determine the affinity of the receptor for its ligand and also to find the number of binding sites. So Stackad plots both the equilibrium constant and the number of binding sites per antibody molecule, or I can say number of binding sites per antibody molecule as its valency can also be obtained. If all the antibodies have the same affinity, the Stackad plot yields a straight line, as you can see in the case of Q over here in the graph. Q is a straight line. This is Q, it is a straight line. So that shows it is a monoclonal antibody or it is homogeneous. The y-intercept in the valence, that is this region. Y-intercept is this. This is y-axis. Sorry, this is x-axis. And it represents the valency or the number of binding sites per antibody. Okay, now for R it is 2 and for Q it is 4. So 2 binding sites are found for IgG and 4 binding sites are found for IgA. So R is IgG and Q is IgA. Now coming to the statements. Antibody Q is possibly a monoclonal while R is polyclonal. That is why you got a straight line for Q and a curved line for R. I already explained that. So A is correct. The curved nature of Stackad plot for R indicates that it cross reacts with the blocking reagent. That is not correct. It doesn't cross react with the blocking reagent. If the antibodies are pooled and have a range of affinities, the scattered plots yield a curved line. That is the curve why you got a curved nature for R, because R is polyclonal. The average affinity of R is more than the affinity of Q to the antigen. That is also correct. Antibody Q is possibly IgA. That is why it is having 4 binding sites, and R is IgG. That is why it is having 2 binding sites. So that is also correct. The valency of the antibodies cannot be inferred from the checkered plots. They can be inferred. That is why 2 and 4, I told you it is for IgG and IgA. That is binding affinity, the number of binding sites or its valency. So the valency can be determined. So E is not correct. So which are the correct statements? A, C and D, and your option 3 is the answer. A is correct, C is correct, and D is also correct. So option 3, A, C, D is the answer. Now next one. In Drosophila, a cross was set between a male homozygous for alleles S plus, you get phenotype A, and a female homozygous for SS, you get phenotype B. S plus is dominant and S is recessive. All of the F1 individual thus obtained from the had the phenotype B. When F1 individuals were crossed among themselves, all the progeny obtained were of phenotype A. The following explanations were proposed for the results obtained. Which of the following option is correct? Now what they have told in the question was, this is S plus which is phenotype A, and this is S which is phenotype B. When they were crossed, you got the F1 generation which is showing the phenotype B even though S plus is dominant. So it is showing the phenotype of the recessive gene, not the dominant gene. That is why B, phenotype B is being seen. Now when this was crossed, you got the F2. So these are the F2 parents. These are the gametes of F2 generation, and these are the offsprings. And what did you observe? The phenotype was of A in the F2 generation. A phenotype you got. This shows that mother's genotype and not the phenotype controls the phenotype of the offspring. That is, mother's genotype control the phenotype of the offspring. Here genotype of mother is in F2 generation S plus S, and S plus is dominant. So all the offsprings will be A. Because mother's genotype is S plus. That is why this SS also is having A phenotype. Only genotype of mother matters, not her phenotype. Then in the first cross, the genotype of the mother is small s. This is the mother. So the genotype of the mother is small s. So the offsprings will be of B phenotype. So this is exhibiting genetic maternal effect. So this is an example of cytoplasmic inheritance. That is not correct. This is exhibiting genetic maternal effect. That is correct. This is quantitative trait. No. This is exhibiting gene interaction with epistasis. No. The trait is showing position effect variegation. No. So the answer here is B only. Answer is option 2. Then next one. The amino acid tRNA synthetases in an organism have evolved to catalyze amino acylation of their cognate tRNAs, at which position they have asked. So in the first step, the amino acid carboxylate attacks the alpha phosphate of the ATP, and synthesis of an amino acyl adenylate AAAMP occurs with the release of inorganic pyrophosphate. That is what happens in the first step. Then in the second step, the transfer of the activated amino acid to either the 2' OH or the 3' OH on the terminal adenosine of the accepting tRNA. So the answer here, first one, either at the 3' OH or 2' OH position of the adenosine at the CCA end. That is correct. Coming to the next question, it is from NMR. Shown below is the proton-coupled carbon-13 NMR spectrum of sodium trimethylsilyl propane sulfonate (DSS), a common internal chemical shift standard used in NMR spectroscopic studies of proteins and peptides. Also shown on the spectrum is the structure of the DSS, in which the different carbon atoms have been labeled A to F. The peaks in the NMR have been labeled 1 to 4. Which of the following represent the correct assignments for the carbon in DSS? The nuclear spin quantum number of H1 and C13 are half. So these are the peaks. This is first, this is second peak, this is third peak, and this is fourth peak. Which carbon atoms do these peaks represent? You have to find out. Now if you see this one, all the carbon atom joined to Si, that is silicon, will be of the same nature, that is A, B, C carbons. A, B, C carbon, they are of the same nature. So peak 4 in the option is A, B, C, this one. So peak 4 is for ABC carbon. So you can write, you see 2 and 3 options. It is peak 4 is ABC carbon. Now we have to see for others. Now in this one, CH2 S double bond O, this carbon, what happens there? So in this group, the proton is adjacent to the strongly electron withdrawing oxygen atom. The proton is therefore de-shielded and requires lesser magnetic field to resonate. This de-shielding shifts the absorption downfield. So peak 1 will be F. That is this. Peak 1 will be carbon F. If you see option 2, it is peak 1 is for carbon F. The answer is option 2. Now followed by, after that comes peak E, and D will be peak 3. So the answer here is option 2.